Good day. You can see we're looking at a sheet that says DNA, structure, replication, transcription, translation, notes. So we're going to throw a lot of notes at you and then we'll look at some PowerPoints that I think will make things even clearer. But this should be straightforward. So DNA controls all the cell activities, tells the cell what to do, when to do it, how to do it, and the information that contains genetic information, or the nucleus contains chromosomes which carry genetic information. Chromosomes are made of DNA, deoxyribonucleic acid. Genes are sequences of DNA that code for certain proteins. And the previous videos or earlier videos, you should have seen that proteins have specific functions. Being human, you have 23 pairs of chromosomes. That means you get 23 from your mother, 23 from your father, for 46. And you'll find 46 chromosomes in every body cell. So you're filling in the blanks as we go. Now we've talked about DNA already, and there's the fancy term, deoxyribonucleic acid, the double helix, the rails of the ladder are the sugar phosphate backbone. And we have the A, T's, C's, and G's, and A and T are complementary, that means they fit together, and C and G are complementary, fitting together. So one big thing to realize, DNA is too big to leave the nucleus. Now that's a problem in the sense the protein is made in the ribosomes and the ribosomes are outside the nucleus in the cytoplasm. So before we get to that, we got to talk about RNA. RNA is ribonucleic acid. So it says similar to DNA, but instead of a double strand, it's a single strand. So it's like having half a ladder that's been sliced down the middle. RNA does not have T's, doesn't have thymine. Instead, it has uracil. So if you ever draw RNA, make sure you don't put any T's in it. They have to be replaced with U's. There are many kinds of RNA, but we're going to focus on messenger RNA and transfer RNA. Messenger RNA makes a copy of a section of DNA in the nucleus and takes it to the ribosomes. Transfer RNA float around the cytoplasm and carry amino acids and bring amino acids to the ribosomes. So we've talked about replication already. Now let's get to DNA making protein. The building block of proteins are amino acids. When amino acids combine, they can make proteins. Where that happens, ribosomes. So we are going to look at processes called transcription and translation. Transcription, the purpose is to make a copy of a section of DNA. The DNA acts as a template, and the messenger RNA makes a complementary copy. So that means, hey, if you have an A, you get a U. If you have a T, you get an A, a G, you get a C, etc. So the DNA has to unzip. The nucleotides will fill in. You've made your messenger RNA, and at a certain point, it'll break off, and it will leave the nucleus, taking its genetic information with it. And the DNA will zip back together. Translation happens at the ribosomes. So now we're taking that information in the messenger RNA and we're going to translate it into proteins. So translation happens in the ribosomes. So the messenger RNA has left the nucleus, it's now at the ribosome. The ribosome reads the messenger RNA as a series of triplet codons. And you say, what's that mean? Well, it reads these A, T, C's, and G's 3 letters at a time. And you will have a codons chart, and we'll look at that in more detail, but we'll just give you a quick glance of it. Let's say I had something that was C, C, A. Okay, well here's a C, there's a C, and I come down to A in here, and I see, oh, that's going to be an amino acid known as proline. So we've got an easier chart than this, but that's what it's saying. This allows us to read 3 letters at a time and see what amino acid is going to be brought in by the transfer RNA. So the codons, it's a 4-character system. So we have these 4 letters to choose from. The bases combine to make 3-letter words. That's why we said triplet codon, triplet meaning 3. So 4 to the 3rd power gives us 64 possible combinations of 3-letter words. But it turns out there's only 20 amino acids. So what you may have noticed here is quite a few things can make serine or tyrosine and whatnot. And it gives a bit of redundancy, and that is actually turns out to be very useful, as we will see. So here's a diagram of what would be going on the ribosome. And so it says, okay, we're moving in this direction. And so right now, we can see we have, this is our messenger RNA. These three letters, UUC, would be an example of a codon, triplet codon. Well, here's our triplet anticodon. This belongs to the transfer RNA. And on top of it, the little ball there represents an amino acid. So we had an amino acid here, and another amino acid has just been brought in. And you can see this transfer RNA is leaving. It's leaving behind its amino acid. And so we're assembling like so. Boom, boom. This amino acid is going to add there. Then this amino acid is going to join there. So we get a very precise sequence of DNA being added together. DNA is the coding strand and a template strand. It has two strands. Coding strand is going to code for messenger RNA. That's where we get the codon. Transfer RNA will have the anticodon and the corresponding amino acid. When we want to know what that amino acid is, though, we have to use the messenger RNA's codon. And like I say, we'll find a better chart to show how that works. And this diagram is just trying to show the sequencing all the way as you go from DNA all the way down to an amino acid and get a protein. We can get a little more exotic where we look at start and stop. Start tells us where to start reading the DNA. Stop's telling us where to stop reading, and that will determine the length of our messenger RNA and ultimately the length of our protein. You will see this if you take Biology 11, and you'll see it again in Biology 12. So it's really great if you get a good handle on it. Here's our final summary. DNA is a template for RNA, messenger RNA. Messenger RNA, small enough, can go to the nucleus, to the ribosomes. Here it will meet transfer RNA that combines according to the codons and anticodons, and it will bring the correct amino acid. And the amino acids will stitch together and will make a protein. So next up, we're going to watch a PowerPoint about the transcription process.